DNA, or deoxyribonucleic acid, has the ability to make genes. Those genes are able to make up your physical features, such as the pigment of your hair and eyes. Now, how is this done, you may ask? Well, you can give thanks to protein synthesis. Synthesis means to make something. So protein synthesis means that the cells within your body are going through a process to create protein. There are two steps to protein synthesis: transcription and translation. Transcription is the process by which the information in a strand of DNA is copied onto a new molecule of messenger RNA, or mRNA. Before we give you the definition of translation, let us go into more detail about the first step of protein synthesis. Transcription takes place in the nucleus of your eukaryotic cells. The differences and similarities between a DNA nucleotide and an RNA nucleotide is that they both have one phosphate and one 5-carbon sugar. However, RNA is a ribose sugar and DNA is a deoxyribose sugar. Both have 4 different nitrogen bases. For RNA, or ribonucleic acid, instead of thymine, uracil is used in its place. DNA is a double helix, while RNA is a helix, so RNA only has to copy one strand of DNA. Now, to be even more specific, there are 3 types of RNA. There's messenger RNA, which copies genetic information from DNA and carries it to the ribosomes. Then there is ribosomal RNA, which is the protein that makes up the ribosomes, and transfer RNA. This transports amino acids to the proteins in the ribosome. There are also transcription factors, which bind DNA sequences, called enhancer and promoter sequences, in order to recruit RNA polymerase to an appropriate transcription site. This enzyme binds to the DNA of the Tata box and separates the double helix. The Tata box helps the enzyme, which in this case is the RNA polymerase, figure out where to bind to this strand. It is a DNA sequence of mostly adenine and thymine. This simple piece is located in the promoter region of genes. A promoter contains an initiation site where transcription of the gene begins. Guanine is put at the 5', and about 250 adenines are put on the 3', called poly-A tail. This makes it easier for the mRNA to leave the nucleus and protects it from getting destroyed by other enzymes and able to connect to organelles later on. RNA then unwinds the DNA from the 5', then starts the process of elongation, where the RNA strands get longer as more and more nucleotides get added on. DNA/RNA hybrid is formed, and it is when one strand is DNA and the other is a complementary RNA strand. This happens because a newly synthesized RNA strand with a DNA template strand, so the RNA polymerase can copy it. RNA polymerase copies DNA sequences downstream of the Tata box towards 5', which copying into a messenger RNA. The message on the mRNA is based off of the DNA original instructions. RNA polymerase connects complementary RNA bases to the DNA. RNA bases bond to form a single strand of mRNA, which includes both introns and extrons. The introns are between each exon and get removed during a process called splicing. What happens here is the introns are taken out of the sequence, leaving the exons in separate pieces. After this, the remaining pieces are combined together, forming the mRNA. RNA continues to elongate until it reaches the terminator, which is a specific sequence of nucleotides that signals the end of transcription. Transcription stops, and mRNA polymerase and the new mRNA transcript are released from DNA. Mutations can occur when a protein that has been made becomes unusable. A form of a mutation is when a single base is changed, and then the mRNA copies that strand, which gives the organism a genetic disorder such as a Tay-Sachs disease. Without transcription taking place, there would not be any mRNA being formed and moving into the ribosome for translation. This means that no protein will be made, which results in the cells not having any building blocks. Translation is the process in which messenger RNA takes information that is passed from DNA and is then turned into a series of amino acid bonds together with peptide bonds. Translation takes place in the cytoplasm. RNA nucleotides consist of one 5-carbon ribose sugar, a phosphate molecule, and 1 out of 4 nitrogen bases: adenine, guanine, cytosine, and uracil. After transcription, the mRNA takes the photocopy of the DNA and leaves the nucleus and goes into the cytoplasm. Then, an initiation complex is assembled consisting of a strand of mRNA, a small subunit of a ribosome, and a charged tRNA carrying methionine. Then, the initiation complex moves across the strip until it binds with the start codon. To finally form the initiation complex, a large ribosomal subunit joins to form the initiation complex. The elongation of amino acids begins, and RNA polymerase adds the amino acids and the additions of new nucleotides. Ribosomes are composed of 2 subunits, one small and one large. Ribosomes match the base sequence on the rRNA in sets of 3 bases with codon to tRNA molecules that have complementary bases with anticodon. The 3 tRNA sites are labeled P, A, and E. The tRNA first binds to the P site. The P site is the ribosomal site most frequently occupied by the tRNA carrying the growing peptide chain. Then, because the P site is a pyrimycin and can link via a peptide bond to the A site, the tRNA goes to the A site. The A site binds to the now amino acyl tRNA, which holds the new amino acid to be added to the polypeptide chain. Finally, the tRNA binds to the E site and facilitates release of the mRNA from the ribosome. The mutation for translation is called nonsense mutation. Nonsense mutation is when an amino acid gets changed to a stop codon, resulting in premature termination of translation. Translation is important because it is the process of creating a protein out of mRNA, which then makes up you.